Hello, everyone, and welcome back to the lecture series on synthetic and natural macromolecules. We have already examined the properties of esters and natural macromolecules based on esters. In this lecture, our focus will be on amino acids and natural macromolecules derived from them. However, before moving on to the main topic of the lecture, let's begin with a brief review of the synthesis and properties of amides. An amide is the product of a substitution reaction between an amine and an ester. As demonstrated in this reaction scheme, the partially negative nitrogen atom of the amine is attracted to the partially positive carbon of the carbonyl group in the ester. As a result, an alcohol is lost and an amide is formed. This reaction follows the general mechanism we discussed for nucleophilic substitutions at carboxylic acids and their derivatives. Amides are relatively stable. However, they can be hydrolyzed in hot water to yield a carboxylic acid, or carboxylate ion, and an amine. Notably, this reaction is significantly facilitated in the presence of acids or bases. Amides are widespread in nature, and some important representatives are shown here along with a brief description of their properties. Nevertheless, the most important molecules based on amides are peptides and proteins, which we will examine shortly. Research at the interface of chemistry and biology, known as biological chemistry, is an exciting and dynamic field for scientists in both disciplines. Biological chemists apply the principles of chemistry to understand the molecular basis of biological processes relevant to medicine, biotechnology, and other life sciences, including structural biology, biochemistry, genetics, pharmacology, and physiology. They then use this knowledge to develop new chemical transformations, materials, or advances in medicinal chemistry. This differs from biochemistry, which aims to understand life in terms of its processes and reactions, but does not necessarily focus on the molecular scale. In this lecture and the following few, I will provide a brief overview of some fundamental aspects of biological chemistry, focusing on proteins, polysaccharides, and nucleic acids. We will examine these macromolecules from a molecular perspective and discuss the significance of nitrogen and oxygen-based functional groups in each case. Proteins are nature's polyamides and are unbranched polymers formed from monomers called amino acids. An amino acid has both its carboxyl group and amine group attached to the same carbon atom called alpha carbon, which is the second carbon atom in the chain. A hydrogen atom is the third group bonded to the alpha carbon, and the fourth is the side chain, denoted here in the generic form as R. In the aqueous cellular environment, the amino and carboxyl groups of amino acids are charged because the carboxyl group transfer a hydrogen ion to water, forming hydronium ions. These hydronium ions, in turn, transfer protons to the amine group. Since this process occurs throughout the solution, it can be viewed as an intramolecular acid-base reaction. Proteins are composed of approximately 20 different types of amino acids, distinguished by their side chains, which range from a hydrogen atom to a polycyclic nitrogen-containing aromatic structure. Proteinogenic amino acids are classified into several subgroups based on the nature of the substituents attached to the alpha carbon. The first group consists of nonpolar amino acids, which have linear or branched alkyl chains, hydrogen, or an alkyl chain containing a sulfide subunit attached to the alpha carbon. Next is a small subgroup that includes amino acids with polar alcohol or thiol subunits connected to the alpha carbon. Following this, there are several amino acids with aromatic side chains. Amino acids containing side chains with amino groups are classified as basic, while those with an extra carboxyl group are acidic. It should not surprise you that the nature of these side chains plays a major role in determining the shape and function of a protein. Each amino acid is linked to the next through an amide bond formed by a dehydration condensation reaction, in which the carboxyl group of one monomer reacts with the amine group of the next. In biomedical sciences, this amide bond is commonly called a peptide bond. Accordingly, The polypeptide chain, or the backbone of the protein, has a repeating structure consisting of an alpha carbon bonded to a peptide group, which is then bonded to the next alpha carbon, followed by another peptide group, and so forth. The various side chains, represented here as R groups, dangle from the alpha carbons on alternating sides of the chain. For instance, in this figure, you can see a portion of a polypeptide chain consisting of glutamic acid, arginine, glutamine, histidine, methionine, aspartic acid, and serine. Each protein has its own amino acid composition, with specific proportions of various amino acids. However, it is not the composition that defines a protein's role in the cell. Rather, the sequence of amino acids determines its shape and function. Proteins range in size from about 50 to several thousand amino acids. Yet, from a purely mathematical perspective, with the 20 different proteinogenic amino acids, even a small protein of 100 amino acids has a virtually limitless number of possible sequences. In reality, however, only a tiny fraction of these possible sequences occur in actual proteins. For example, even in an organism as complex as a human being, there are only about 100,000 different types of proteins. A protein folds into its native shape as it is being synthesized in the cell. Some shapes are simple, forming long, helical tubes or undulating sheets. Others are far more complex and include baskets, Y-shapes, spheroid blobs, and countless other globular forms. Biochemists typically define a hierarchy for the overall structure of a protein, which we should briefly examine here. The primary structure of a protein is its most basic level of organization and refers to the sequence of covalently bonded amino acids in the polypeptide chain. The secondary structure refers to sections of the chain that, due to hydrogen bonding between the carbonyl oxygen of one peptide group and the hydrogen atom of the NH subunit of another nearby peptide group, adopt shapes called alpha helices and beta-pleated sheets, as shown on the right. The tertiary structure refers to the three-dimensional folding of the entire polypeptide chain, which results from a combination of mostly intramolecular forces. In many proteins, characteristic folding patterns form regions that play a functional role, such as binding a hormone, attaching to a membrane, or forming a membrane channel. Finally, the quaternary structure occurs in proteins composed of multiple polypeptide chains and refers to the way these chains assemble. This is the most complex level of organization in proteins and is typically based on a wide variety of intermolecular forces common in multi-subunit proteins. Hemoglobin, for example, consists of 4 subunits, as shown on the right. The key point in protein organization is that only the primary structure relies on covalent bonds, whereas the secondary, tertiary, and quaternary structures depend primarily on intermolecular forces, which you learned about in the first module of this course. Two broad classes of proteins differ in the complexity of their amino acid compositions and sequences and, therefore, in their structure and function. The first class includes fibrous proteins, which have relatively simple amino acid compositions and correspondingly simple structures. They are key components of hair, wool, skin, and connective tissue, that is, materials that require both strength and flexibility. Like many synthetic polymers, these proteins contain a small number of different amino acids arranged in a repeating sequence. As an example, let's take a look at collagen, the most abundant animal protein, comprising up to 40% of the human body's total protein content. In collagen, more than 30% of the amino acids are glycine, while another 20% are proline. It consists of 3 polypeptide chains, each forming an extended helix that wind around each other. The peptide carbonyl groups in one chain form hydrogen bonds with the peptide NH groups in another, resulting in a long, triple helical cable. As the main protein component of tendons, skin, and blood vessel walls, collagen has high tensile strength. For instance, a collagen strand 1 millimeter in diameter can support a weight exceeding 10 kilograms. Another example of fibrous protein is silk fibroin, which is secreted by the silk moth caterpillar. Glycine, alanine, and serine make up more than 85% of its amino acid composition. The fibroin chains run parallel to each other and form interchain hydrogen bonds, creating a pleated beta sheet structure. Stacks of these sheets interact through dispersion forces, giving fibroin its strength and flexibility while limiting its stretchability, making it perfect for a silkworm's cocoon. Globular proteins have much more complex compositions, often containing all 20 amino acids in varying proportions. As the name implies, globular proteins are typically rounded and compact, exhibiting a wide variety of shapes and functions. They can act as defenders against bacterial invasion, messengers that trigger cellular responses, catalysts of chemical reactions, or membrane gatekeepers that regulate cellular concentrations, among many other roles. The locations of specific amino acid side chains are crucial to a protein's function. For example, in catalytic proteins, enzymes, a few side chains form an active site, a crevice that closely matches the shape of the reactant molecules. These groups typically hold the reactants in place through intermolecular forces, facilitating their reaction by bringing them together and inducing conformational changes that weaken or stretch specific bonds. For instance, ribonuclease, an enzyme found in cows, hydrolyzes RNA. It is a relatively small protein consisting of 124 amino acids and stabilized by 4 internal disulfide bridges. The table on the right provides the amino acid composition of this enzyme. Of the 124 amino acids, 48 are involved in maintaining the protein's structural integrity and stability, while the remaining amino acids have functional side chains extending outward. These include amino, hydroxyl, and carboxyl groups, among others. However, despite the presence of many reactive functional groups, only a few play a direct role in catalysis, likely just 2 histidine residues and 1 lysine residue. In this figure, you can see a portion of the ribonuclease structure surrounding one of its catalytic amino acids, histidine at position 12. This sequence consists of 7 amino acids, each with a unique functionalized side chain. Among them, 6 distinct functional groups are present, including 2 carboxyl groups, an amide, a guanidinium group, an imidazole group, a sulfide, and an alcohol. While all of these groups have potential chemical reactivity, only the histidine residue is actively involved in catalysis. Experimental studies have shown that even a slight modification in one of these critical side chains can dramatically reduce the enzyme's catalytic efficiency. This finding reinforces the fundamental principle that a protein's amino acid sequence determines its structure, which in turn dictates its function. To sum it up, in this lecture, you were introduced to one of the most important families of biological macromolecules, proteins. You learned that proteins are polyamides formed from up to 20 different types of amino acids, known as proteinogenic amino acids. These amino acids differ from one another by their side chains, which are attached to the alpha carbon atom. We then discussed the relationship between protein structure and function. You now know that fibrous proteins have elongated structures and primarily serve structural roles. In contrast, globular proteins have compact shapes and are involved in metabolic, immune, and hormonal functions. The key idea to remember is that a protein's amino acid sequence determines its shape and function. In the next lecture, you'll be introduced to sugars and polysaccharides. Thank you for your attention.